So here the introduction. Carica papaya, commonly known as a papaya, is tropical fruit rich in bioactive compounds such as phytophenols, flavonoids, and enzymes. Extract from the different parts of papaya, including leaf, peels, and branches, have demonstrated potential in the green synthesis of magnetic ferromagnetic. This extract acts as a reducing agent and capping agent, facilitating the formation of nanoparticles while minimizing the environmental impact. In this presentation, we explore the sustainable synthesis of iron nanoparticle using carica papaya extract. We discuss the characterization of these nanoparticles through SEM, FTIR, and also EDS to evaluate their morphology and elemental composition. Additionally, highlighting the superior performance of CPEB, which is branches in terms of iron and oxygen content.
Our background of study for our project is nanoparticles, particularly those smaller than 100 nm have garnered significant interest in environmental and medical applications due to their unique properties. Nanoscale magnetic particles, for example, are notable for their potential in environmental studies and targeted drug delivery. Their chemical behavior and applications are influenced by size, shape, and tendency to aggregate, with smaller particles exhibiting increased viscosity, melting point, thermal conductivity, and antimicrobial activity. Green synthesis, an eco-friendly method using biological extracts like plant materials, is preferred for producing iron nanoparticles due to its affordability and reduced toxicity. This method prevents aggregation and oxidation, enhancing the stability of nanoparticles. Papaya leaf extract specifically has been highlighted for its antiviral, anti-diabetic, anti-cancer, and anti-inflammatory properties, demonstrating the potential of green synthesis in producing biologically active and environmentally sustainable nanoparticles. Our objective of these projects is green synthesis of magnetic nanoparticles using iron 3 chloride extract of papaya leaves, peels, and branches. Second, to produce the synthesis of nanoparticles with using the different part of carica papaya as reducing agent. The last one, study the characterization of magnetic nanoparticles from papaya leaves, peels, and branches as reducing agent.
In the methodology session, I will explain how to conduct the experiment of extraction the carica papaya peel, leaf, and branch, as well as the synthesis of magnetic nanoparticles. So the first step in extraction of carica papaya, peel, leaf, and branch, we need to peel off the papaya fruit and chop the branch. Also need to chop the leaf into a small piece. After that, we should wash every part using a deionized water. Next, for the step 2, we should dry each part in the oven at 60 degrees Celsius for 1 day. After 24 hours, we will grind each part into a dust form. After the grinding process, we will insert each part of powder into the bottle and label it. After that, we will weigh each part of the powder 10 grams for the extraction process. In extraction process, we need to mix the each part of powder with 100 ml distilled water using the hot plate and magnetic stirrer at 700 rpm and 70 degrees Celsius. After the extraction process, we need to filter the solution and then we'll cover the beaker with the aluminum foil and store it in the refrigerator at 4 degrees Celsius. So for the synthesis process, we need to weigh 2.7 gram iron trichloride and dilute it in the boiling flask with adding with at 100 milliliter distilled water for and stir it for 30 minutes at 700 rpm. After 30 minutes done, mix the iron chloride and extraction solution. We continue stir the solution for 24 hours and cover the beaker with aluminum foil. After 24 hours stir the solution, we need to neutralize the solution to pH to 7 with adding the sodium hydroxide solution. After done the neutralization process, we need pipette the solution into the centrifuge tube, and then we need to centrifuge the solution at 1000 rpm for 10 minutes. After that, we need to discard the excess water for the dry process. After 24 hours of drying process, the magnetic nanoparticle will appear in solid form in centrifuge. Then we need to insert the magnetic nanoparticle into the tube glass for lab test.
So here the first characterization of iron nanoparticle, which is FTIR. As you can see, the x-axis represents the wave number, while the y-axis shows the percentage transmittance or absorbance. The graph contains color-coded bands indicating specific functional group, which is OH stretch hydroxyl around 3500 till 3200 in orange. CH stretch, which is alkyl, around 3000 till 2850 in yellow. C double bond O stretch, which is carbonyl, around 1750 till 1700 in green, and asymmetric stretch around 1650 till 1550 in cyan, and ferrum oxide stretch around 500 and 550 till 500 in pink. So in summary, while all the three samples contains the same functional group OH, CH, C double bond O, asymmetry, FeO stretches, the variation in peak intensity and sharpness suggests a difference in the concentration and the structural environment of these functional groups. Sample B, which is branch, generally exhibit the strongest peaks. The second characterization of iron nanoparticle, which is scanning electron microscopy, SEM. SEM image indicate a relatively uniform size distribution with an average particle size of approximately 25 microns. So the first sample, which is branch, the nanoparticle synthesized using the CPEB extract, where a similar spherical shape and such uniformity are present, with some degrees of agglomeration likely due to the magnetic nature. The second sample, which is pale, nanoparticles synthesized using the CPEP extract exhibit a predominantly spherical morphology with a smooth surface. And the last sample, which is leaf, present the nanoparticle synthesis using the CPEL extract display a thin organic layer on the surface of the nanoparticles, indicative of the effective functionalization that enhance their stability, dispersibility, and biocompatibility. The last characterization of iron nanoparticle, which is EDS, that show all the element in a sample. So the first sample, which is a peel extract, present of iron, magnesium, calcium, sodium, oxygen, and carbon. The presence of iron and oxygen is confirmed the formation of iron oxide. Magnesium, calcium, and sodium are likely derived from the mineral content of the papaya peel, while carbon is attributed to the organic capping agent from the peel extract. So the second sample, which is leaf, all the elements are present, but the absence of magnesium and the reduce of oxygen content may be due to the lower mineral content in the leaf extract compared to the peel and branch extract. The detected elements still indicate the formation of iron oxide. And the lastly is branch. Branch extract present of all the same element, an additional element which is chlorine. The presence of chlorine could be due to the residual chlorophyll or other organic residual compounds present. The higher concentration of iron and oxygen suggests a more efficient synthesis process, resulting a higher purity of iron oxide. Nanoparticle. The element magnesium, calcium, sodium, and carbon originate from the organic and mineral content of the branch extract.
Magnetic nanoparticle benefits to society are medical application. Magnetic nanoparticles synthesized through green methods are often more biocompatible, making them safer to use in medical applications such as targeted drug delivery, magnetic resonance imaging or MRI, and hyperthermia treatments for cancer. Next, reduce toxicity. Green synthesis uses natural non-toxic materials and processes which minimizes environmental pollution compared to conventional chemical synthesis method. This is crucial in reducing the ecological footprint of nanoparticle reduction. Lastly, environmental remediation. Magnetic nanoparticles can be used for water purification and treatment of contaminated sites, including oil spills, cleanup and removal of heavy metals or pollutants from water, contributing to environmental conservation and public health. In conclusion, the magnetic nanoparticles that use iron 3 chloride hexahydrate that use the process of green synthesis with the extract of papaya leaves, peels, and branch have indeed come to success. This is stated because of results from FTIR, SAM, and EDX tests that stated that the ions of Fe, which are iron, indeed exist in the nanoparticle tested. This shows that the nanoparticle tested indeed contain magnetic force because of the Fe ions in the nanoparticles. This experiment shows that in producing magnetic nanoparticles, the lowest toxicity rates were observed because different parts of carica papaya played the role of reducing agent to reduce toxicity. It also shows that in the results, each part of carica papaya stated different results which classify the characteristic of one another.